When I first joined Professor Nicole Swan's lab, I had no idea what was going on. I knew so little that I was actually ignorant of how ignorant I was. The Swan lab uses large data sets of electrical brain activity to better understand human movement. However, when I first came in, I had never taken a coding class. I really couldn't tell you the difference between an array or a matrix. Knew nothing about electricity, couldn't tell you the difference between a volt or an amp. And I hadn't taken a single neuroscience, let alone psychology course, up until that point. Needless to say, I had a lot to learn, and I had to learn it fast. So we primarily use electroencephalography. That was actually my first challenge, figuring out how to pronounce that word, and hopefully I give you a good pronunciation spell out on the slide. And so we use electroencephalography, EEG, to record the electrical activity from people's brains from the surface of their scalps. Simple enough. However, when looking at these data, we can see on the top row we have this nice long signal, and then with the red box zooming in on it, we can see it's quite complicated, it's quite messy, but thankfully electrophysiologists have designed methods to understand these data. However, it was now my challenge to understand their methodology. So before we continue, I just have to give a screenshot of a generic MATLAB documentation. MATLAB is the language that I've learned to code in, just because many hours have been spent here reading and learning, because before learning any of the methodology of the signal processing you want to apply to EEG data, you also have to know how to tell the computer how to do that, how to code it. So moving right along, the first problem that we find with EEG data is blinks. So when you close your eye, your eyeball literally goes up into the back of your head, and because your eye is positively charged, and if you have electrodes on your scalp, every time you close your eye, you increase the polarity, and that can be read by EEG electrodes. So looking at the left graph here, we can see, ignore these acronyms, these are just various EEG electrodes, and we can see that when someone blinks, indicated by this EOG, which is an eye electrode, we can see an increase there. We can also see an increase across many channels, but we're not interested in studying eye activity. We're interested in understanding brain activity. So ICA, or independent component analysis, gives us a powerful tool to overcome this problem. But when I first was learning about ICA, it's a mathematical procedure that's a bit over my head, and so I had to kind of pull back and realize I didn't need to understand every little detail of how ICA worked, but that I could use ICA to, now looking to the figure on the right, to remove this hump in the data, the eye blinks that are artifactual and not interesting. Here I learned that I didn't need to get into every single detail, but using the systematic magic of ICA, I was able to get a cleaner data set. But EEG data is also plagued by muscle artifacts. So when people move their faces or necks while you're recording them, that also creates fluctuations. And now this is a much larger channel plot. And the highlighted green area, if we look specifically kind of near like that maroon dark splotch, we can see a lot of activity where it's going much higher and lower than it is around it. And we can, using kurtosis, which is a measure of how sharp a distribution is, and Extrema, which is just a maximum or a minimum, we could remove parts of the data that we defined as to be artifactual. Basically, they had very high kurtosis or very high extrema. But again, this is a process where I'm able to use functions made by someone else in order to better understand the EEG. And the great folks at EEG Lab have an aptly named EEG wiki where I would learn to do lots of this, and being able to use the work someone else has done in order to better understand your data saves you, the researcher, time. And this is something that took me a while to learn because I really wanted to understand every nitty-gritty detail of what was going on. But eventually I came to see that if I spent so much time trying to understand every aspect of my analysis, I would never get it done. I just needed to understand what my results meant. And now that gets me to my next part. So when understanding EEG, it's important to understand the frequency domain. But when I started this part of my analysis, I couldn't explain to you what in the world the frequency domain even was, and I hit quite a roadblock because I had this vague idea of what I wanted to do, but I had no way of implementing it. So under the guidance of my PI, I was able to take this online course where I was able to get a good grasp on what the frequency domain was and how it can be important in understanding my results. So now answering the question, what in the world is the frequency domain? So to the left, we have simulated data of just a sine wave fluctuating over time. And what the Fourier transform does is lets you take data in the time domain to the left into the frequency domain on the right. And we can see that since I made this sine wave to be at 20 hertz, that the entire frequency domain shows it to be at 20 hertz. So that's handy. But how is this helpful for understanding EEG? So now looking at waves at different frequencies, we can see they have different frequencies in their frequency domain. But more importantly, we can see this red and blue wave. If we added them together, we would get the purple wave. And this is where the frequency domain starts to be more helpful. Because I'm not sure if the video is covering it, but we can understand how multiple frequencies contribute to our time series data. So now taking our much more complicated EEG signal that we started with and putting that in the frequency domain, we can better quantify it and understand it. It gives us a new variable to understand the EEG. And taking this a step further, with amplitude being on the left of the top graph, telling us how much that a given frequency contributes to the overall signal. But this is only a summary over one moment in time. What would be more handy is to see how the frequency domain changes over time, not just a single snapshot. So taking our amplitude value and now making that based on a color, we can look at how frequency changes over time. This is more or less known as instantaneous frequency. And this online course that I mentioned before was the only way I was able to wrap my head around these ideas and understand a bit of the math. And here it was more important for me to slow down and really grasp the mathematics behind what was happening, because without it, I couldn't interpret my results. I couldn't tell you what they meant. So here it made more sense to slow down. And eventually, with these signal processing toolboxes under my belt, I was able to ask and answer an experimental question, which I won't labor too much here, but looking how when people are making a slower movement, when they're less certain, they're more hesitant about that movement, how the frequency time domain changes with respect to the amplitude. And so here's a very similar plot to before, and we can see in the highlighted red region here with the big arrow, we can now see differences between different experimental conditions. And now the kind of final step of where I'm at in my data journey is before we had those nice time frequency plots, but in order to understand which of those results are statistically significant, that's its own challenge, because as we saw before, there were over 1,000 time points running across time, and then many time points running up in the frequency domain. And now that gives us 30,000 or tens of thousands of time frequency points to compare and cross experimental conditions. In other words, we're trying to run 30,000 t-tests, and running that many t-tests, you're bound to get false positive results. And so currently, I'm understanding how utilizing non-parametric statistical testing can be a powerful way to negate this high false positive rate issue with the EEG. And again, this is just a screen grab from one of the more complicated titled papers I had to read. But throughout this process, I really learned how, in some cases, like with ICA and the artifact rejection, that a conceptual understanding sufficed, and spending too much time on this, getting caught in the details, I would lose the big picture. Or in other cases, when it came to understanding the frequency domain, this was quintessential to my analysis. I needed to be able to explain what my results were and how to generate them. And taken together, this whole experience has showed me when I need to slow down and better understand the problem at hand. And if a conceptual understanding will work, just go with that. Essentially, I learned how to learn, and now I'm continuing to make sense of my EEG results. And as we started this presentation, we must end this presentation with another honor to MATLAB documentation, because throughout the way I also was able to learn and struggle with the coding documentation.